Thanks everyone for joining us today. I hope you're excited about this talk as I was in preparing it. And I guess we can just get right into it. So what we're going to be talking about today in this lecture is how we got from this European or Middle Eastern or Near Eastern wildcat. It was a bunch of different species that contributed to these moggies that we know and love today with these special particular breeds, but also just the regular short hair cats, the moggies that we love and we have in our lives. So let's get into what we're talking about today. So just to give everyone a few disclosures, I am a biologist by training. My research area is on the mechanisms of scar formation in chronic inflammatory diseases, particularly in the lung. So just with the proviso here, I am not a professional geneticist, so I don't actually teach genetics, but cat genetics is my pet project. So I am a senior lecturer in biomedical science at Aston University in Birmingham. And if you have any questions about this talk, or if you'd like to get some of the resources I use to support what we talked about today, please do email me at j.johnson1@aston.ac.uk, and I'd be more than happy to share these materials with you. I am a devoted cat mom with 2 rescue cats, Darwin, who was born in London in about 2012, and Toby, who was born in Birmingham in about 2014. And they had a big brother, Tiger, who passed away at the ripe old age of 21 after being a pub cat in London for 15 years. And we were actually his retirement plan. He and Darwin were best friends. And when we decided to move to Birmingham, the pub let us take Tiger with us so that he could retire in a nice garden with his best friend. I'd also like to mention that I do not breed cats. And some of the genetic variants we're going to talk about today do have some deleterious effects. They can cause some health problems in cats. And I just want to stress that when talking about these different breeds of cats, my primary concern is for the health and the well-being of the cats themselves. So where did all of these cats come from? So in the United Kingdom, about 26% of adults have a cat. And the estimated population, the feline population of the UK, is 10.9 million cats, and that was from the PDSA told us that in 2020. The domestic cat is known as Felis catus, and it is the only domesticated species in the family Felidae. So this is the Felidae shown in the picture here, and you can see how these different types of cats are related to each other, and when they diverged from each other evolutionarily way back in time. So the genus Felis, which is where our beloved moggies live, they diverged from the other Felidae around 6 to 7 million years ago. And here's a more detailed image of that, just to give you an idea how long ago the earliest cat ancestor was. That was 10.8 million years ago, back in the Middle Miocene era. And we've had a huge amount of diversity in cats since then. So we have 37 species of cats that currently exist in the world. Most of the large cats are in Panthera. They're up at the top, so that includes tigers and lions and jaguars and leopards and snow leopards. And then the cats get a little bit smaller as we go down the list. And then you can see further down here the lynx. There's a lot of lynx and puma in my homeland of Canada. We have the Asian leopard cat, and the domestic cat is down here at the bottom. So you can see how closely related the domestic cat is with the wildcat and some other wildcats that are available or exist around the world, particularly the jungle cat. That's a really interesting one as well. So the domestic cat, Felis catus, is the only one that is currently domesticated, though there is evidence from China about 5,000 years ago that there was domestication of the Asian leopard cat as well. But none of these cats exist in the present time. So how our domestic moggies arose is really interesting. So if you look at this map here, we're mostly focused on this area where the green, Felis sylvestris sylvestris, and the orange Felis sylvestris libica, that area where they overlap. So we think that this is generally where the first cats arose. So there's a lot of evidence of cats, of course, in Egypt a long time ago, but the earliest cats that interact with humans were actually a little bit before then. So it's generally thought that Felis sylvestris sylvestris and Felis sylvestris libica are the genetic precursors to the domestic cats that are around today. And these wild cats can still interbreed with our domestic cats, and this can be a bit of a problem in certain places of the world. One example is in Scotland, where the Scottish wildcat, because they're so closely related to domestic cats, there's quite a bit of interbreeding, and there's a big worry that this species will eventually go extinct, not because there are so few cats, but there are so few purebred Scottish wildcats left. So the first cats that we have evidence for that interacted with humans were in the Fertile Crescent, around the times when the first farms were established, and there's a good reason for that, because stored grain attracts rats and other types of vermin, and cats eat rats. So it was a really nice symbiotic relationship between humans and cats under these circumstances, that we were helping each other. So it wasn't quite domestication, but it was certainly cooperation and cohabitation. And the earliest archaeological evidence we have for this is from Cyprus around 9,500 years ago. And the citation for these papers are at the bottom of the slides if you want to go back and refer to the original publication presenting these results. So there were actually two waves in which the domestic cats spread into Europe. Very early on in the Neolithic period, about 9,000 years ago, we had a spread of cats from the Fertile Crescent into Eastern Europe, predominantly in Romania and Bulgaria. So there's some really interesting archaeological evidence there. But the cats really took off in terms of taking over the world in the classical period. So we certainly have that revered cat in Egypt, and the Egyptians then shared their reverence for the cats, and they spread throughout the old world. And there's a lot of evidence that domestic cats were dispersed along the trade routes that humans set up. The Vikings and the Romans in particular were really good at bringing cats with them wherever they went in the world. And in fact, it was required for a Viking longship to have a cat on board to keep the rats and mice under control. And actually, just as a little bit of trivia, it was the Romans who initially brought cats to Britain. So more into how cats developed and evolved in Europe. The first archaeological evidence we have for domestic cats is in Poland. That was from about 4,000 to 2,000 years ago. And you can see on the map where these cats were found in Poland. They were actually really good evidence that they were living among humans during this time as well. You can see on the map that this is close to the area of the wildcats. And it was found that these cats were synanthropic. So there was an analysis of the minerals that were stored in the bones and the teeth of these cats. And they could see from this mineral mix that the cats were eating the vermin that were feeding off of the human stored grain. So there's some really interesting molecular evidence to show that cats were eating vermin and protecting human agriculture from being predated on by these pests. So really helpful symbiotic relationship between humans and cats. So finally, in the Middle Ages, that's when we started to get variations in coat color and people breeding cats to look in a specific way. So the blotched tabby cat, so those of you who caught here a little bit early would have seen a cat, Toby, who is a blotched tabby cat. So it has those thicker stripes in contrast to the mackerel cat, which is the more ancient version of the tabby cat. So you can see in that picture there, a mackerel cat in a bit of Egyptian artwork. So this blotched appearance of tabby cats didn't actually show up until the late Middle Ages, so after the year about 1300 AD. And then that particular genetic variant became a little bit more common. And now this is what we have. So we have a huge number of cat breeds. I'm just going to draw your attention to the diagram that's shown on the right side of the page. So this is called a dendrogram, and it shows you how closely related different, in this particular instance, different cats are. So the branches of the dendrogram show that the cats are related to each other. So for example, if we look at the wildcat here, Felis sylvestris is closely related to Felis caffra, but less closely related to Felis tristami. So you can see that Felis caffra branched off more recently than Felis tristami. So you can see the genetic relatedness of all of these different cat breeds that have developed over the years. So in this particular study by Lipinski et al. In 2008, they looked at the genetic data of over 1,000 cats, and they revealed the existence of 17 different random bred populations. This breeding happened over 5 different continents, And it's left us with about 22 different breeds of cats today. So we'll talk about some of them, but unfortunately, we don't have time to talk about all of them today. You can also see in the dendrogram that there are different colors. So we have the Mediterranean basin in that kind of purplish blue color over here, including things like the Turkish van and the Egyptian Mau. We have the European/American group, which includes cats like the Abyssinian, the Sphinx, and the Maine Coon, shown in red. And we have the Asian cluster, which is shown down here with cats like the Siamese and the Burmese. So you can see that there was geographical differentiation between the different cat breeds that arose over time. And most of these breeds were derived from cats that were already indigenous in that particular area. One really interesting exception to this is Persian, which genetically is actually closer to European and American cats than to Asian or African cats, which I think is really interesting. All right, so how did we get all of these different breeds of cats? And the simple answer is mutation. So hopefully everyone's familiar with the concept of mutations. Not everyone turns into Spider-Man, but in cats, a lot of mutations have arisen. So there are about 70 different mutations or genetic variants that have been defined in domestic cats, and about 40 of these affect the appearance of the cat. So most of them affect coat color, pattern, and length, but there are some genes that regulate the eye color of the cat, as well as the body structure, and we'll get through most of these as we go through the talk today. So just to give everyone a bit of a grounding on the basics of cat genetics. So cats, like humans, are diploid, which means that they have 2 copies of each chromosome. So one chromosome came from mom and one chromosome came from dad. So they possess 38 chromosomes in total. That includes 18 somatic pairs. So those are the ones in that picture that are labeled A through F. The somatic pairs are the ones that are the non-sex chromosomes, and they are equally inherited by males and females. Whereas the sex chromosomes, just like us, we have an X and a Y chromosome. So this particular image, which is called a karyotype, is showing a male cat because there's an X and a Y chromosome. So cats have roughly 20,000 genes in their genome, which is fairly similar to the human genome, which is about 20 to 25,000 genes. But cats, their chromosomes are a little bit bigger. So they have about as much DNA as we do, but divided into a smaller number of chromosomes. And because of this, Down syndrome does not exist in cats. So even if a cat has 3 copies of its smallest chromosome, having that extra amount of DNA is so overwhelming to the system that it's actually embryonic lethal, and those embryos never fully develop into adult cats. So no Down syndrome in cats, which I think is also fairly interesting. So there's a little bit more of a detailed image of the cat genome. So this is showing the different cat chromosomes and the location of different chromosomes on those, locations of different genes on those chromosomes. So the gene name is given in the capital letters, and the location of the gene is shown here. So Q is the long arm and P is the short arm of the chromosome. So we have a pretty good idea of where a lot of these interesting and important genes in the cat are located on their chromosomes. But there's still a lot of research going on in this particular field, so please do watch this space over the next few years. There's lots of really interesting things going on. So just to go over the basics of somatic gene expression and how different genes are expressed. So hopefully everyone is familiar with Gregor Mendel and his experiments on peas, in which the rules of inheritance were worked out. So again, this is specifically talking about somatic chromosomes, so the non-sex or autosomal chromosomes, and the genes that are expressed on these chromosomes are generally considered either dominant or recessive. So if you look at the picture there, this one is about the color of the peas. In this particular state, the yellow is dominant and the green is recessive. So if you cross a yellow and a green, because yellow is dominant, in the next generation, that F1 generation, you only see yellow peas. But if you cross those F1, those heterozygotes, then you can get green peas in the subsequent generation, that F2 generation. So you need to have 2 copies of a recessive gene to be able to express it. Now, keep in mind, there are extra layers of complexity that we're not really going to get into today. There's a theory, well, a concept of polygenic traits, so multiple genes leading to the expression of a single trait. So eye color is a really good example of this. There's also instances of incomplete penetrance, which basically means that a dominant gene isn't necessarily expressed at its full potential all the time. It can be expressed at varying levels. There's also the concept of epigenetics, in which gene expression has an extra layer of control within the cell. And of course, environmental cues can also play a role in how genes are expressed. So in order to work out the relationships between different genes, you can use something called a Punnett square. And this was named after Reginald C. Punnett, who devised this approach in 1905. And this is a simple Punnett square for the color of the peas. In this case, we have the dominant green in this particular case, and recessive is yellow. And you can see that if the green gene is present, it is expressed as a green pea, and you need both copies of the recessive yellow gene in this case to get a yellow pea. And you can get even more complicated by adding extra genes. So this is what a Punnett square looks like for two different traits. So this is both color, so yellow or green, and the texture of the pea, so whether the surface is wrinkled or round. So this is just to give you an idea of what these different Punnett squares look like, and we'll see a couple of these coming up in the next few slides. So we also need to cover the basics of genetic terms you need to know. So I'll be mentioning these terms throughout the talk, so it's good for everyone to be on the same page. So a gene is a trait a gene for a trait is made of DNA. So it is a sequence of DNA in the genome of the animal or plant that you're talking about. The DNA is translated ultimately into a protein, and it's usually the protein that mediates what we observe as the trait. So in the case of a black cat, the protein we observe is called eumelanin, and that basically makes the fur black. A locus refers to a specific location on a chromosome. So that image that I showed you earlier of the detailed cat karyotype, that locus is showing where those different genes are located on specific chromosomes. An allele is a given DNA sequence for a gene, and this is where mutations or variants arise. So there can be multiple alleles or variants of a particular gene, and how those alleles are expressed depends on whether they are dominant or recessive. So a homozygote is an animal or plant that expresses two copies of the same allele. So it may, in the case of a black cat, express two copies of that black pigment, that color pigment that we're going to talk about later. A heterozygote, on the other hand, expresses two different alleles for a gene. So it may be a dominant and recessive, one copy of a dominant allele and one copy of a recessive allele, as an example. So please remember that dominant alleles are observed when one copy is present, and for recessive alleles to be observed, they generally need to be present in two copies. So one example that I've got here up on the screen is the Agouti gene, which is responsible for the banding of color in cat fur. The dominant wild type, so the one that gives cats its stripes, is given the letter A, a capital letter A, and the recessive variant is shown as a lowercase letter a. And this isn't 100% always the case, but generally the dominant one is shown as an uppercase and the recessive one is shown as a lowercase. So please remember, for a recessive trait to be observed, it needs to be present in two copies. All right, so hopefully everyone is ready to go for our deep dive into the genetics of cats. So I'm starting off with black, blue, and chocolate cats because they're a fairly straightforward place to start. For all of these slides where I'm showing pictures of cats, the ones that have names attached to them are mostly from the King's Heath Cat Club. So that is the cat club in my neighborhood in Birmingham. And I put out a call to the King's Heath Cat Club asking for pictures of their cats, and they responded quite impressively. So thank you everyone from the King's Heath Cat Club for providing me pictures of your moggies. Pictures that don't have a name attached to them are mostly just scooped from a Google image search. So the black color of cat fur, so shown really well there by Dexter and Charlie and Bagheera, is mediated by the brown gene. So brown, you can just think of black as being a very, very dark brown, And this is generated by the protein eumelanin. So eumelanin is the true melanin, that black pigment that we see in cat fur, but it's also found in human hair as well. So the brown gene, the dominant one, is the uppercase B, and there are two recessive variants, the lowercase b, which makes a chocolate cat, which is shown down here in the bottom right, and the lowercase bl, which makes a cinnamon cat, which is shown this one right here in the middle. That lighter colored, very cute little kitten is a cinnamon cat. So the color of black and chocolate and cinnamon can also be, oh, hello, Darwin, can also be modified by the gene dilution. It's also known as the dense pigment gene. And dilution encodes for a protein called melanophyllin that essentially dilutes the amount of melanin that's in the cells that are producing the color in the fur. So in this case, a black cat will appear blue. So if you think about a British Shorthair, for example, that blue coat that appears gray is a dilution of a black cat color. Chocolate becomes lilac and it appears light brown. Cinnamon becomes fawn, and ginger becomes cream. So you get a whole bunch of variations of cat color. So in terms of the solid color of cats, you can go from this very, very pale cat here to the fully black cat that we all know and love. All right, moving on to tabby cats. So this is where we start to see a pattern in the fur. So instead of that, that black pattern that we saw in most of those cats, that solid color, in this case, with tabby cats, we get those really interesting stripes. So the tabby gene, which is designated as a T, this accounts for the tabby stripe pattern seen in domestic cats, and it encodes for a protein called transmembrane aminopeptidase Q, or TACPEP. And there are a couple different variations of TACPEP, and we'll go into the details of those in a minute. But the dominant allele, which is an uppercase T and an uppercase M, produces mackerel tabby. So hopefully everyone remembers that mackerel tabby that was in the Egyptian art. And then a recessive allele, so lowercase t with the superscript b, this arose in the Middle Ages, and that produces a blotched tabby. So you get these different patterns of tabby stripes. So I've got mackerel tabbies on the left here, so you can see how thin and aligned these stripes are. I think Tabitha shows that particularly well in this picture. And we also on the right have the blotched tabby. So you have that more conglomeration of the stripes, and you get bigger bands of color of those stripes on the tabby cat. But for both of these types of tabby, we would expect to see an M-shaped stripe on the forehead with thin pencil lines on the face and black eyeliner, which I think most of the ladies are incredibly jealous of, that they get their eyeliner just right. Tabby cats generally have pigmented lips and paws and a pink nose outlined in a darker pigment. They have banding of stripes on their legs and tails. So you can see on Tiger over here, you can see the stripes on his arm there as he's lounging in the grass. And as I mentioned, these stripes come in two different variants, which are mackerel and blotched. And there is a particularly good article in the New York Times that I've given the link to down there if you want to do some further reading on this. So the molecular reason for why this happens is because the underlying skin below the fur is thicker in the areas where the stripes are and thinner where the areas don't have stripes. So hopefully you can see this. And this particular image is a cross section of the cat's skin. And hopefully you can see that lines up with the black bars. The skin is thicker in that area. Where there are yellow bars, it's a little thinner, and then it alternates back to a thick band. And this is mediated by a loss of function mutation in TACPEP that leads these stripes to become essentially less organized. And you get these big blobs and swirls of color in the blotched tabby cat. And again, there's a reference to the original paper providing these this data at the bottom of the page. So another really important gene in the tabby cat, instead of defining the stripe pattern on the whole cat, so that mackerel versus tabby pattern, the agouti gene determines the striping pattern on individual hairs on the cat. So if you look at the picture down here on the bottom left side of the page, the domestic cat has 3 different types of hair, and anyone who has brushed their cat is probably well familiar with what these hairs are like. So at the very bottom part of the fur closest to the skin, we have the very soft down hair, and different cats will have different proportions of these different types of hair as well. So Toby, for example, has a lot of down hair. He has a very, very thick coat. The next largest hair is called the on hair. That's sort of moderate in length. And the longest ones are called the guard hair. So if you've got a cat handy, take a look at their fur and see if you can differentiate the down hair from the on hair and the guard hair. And the Agouti gene has this role of causing the stripe to form on the individual hairs on these cats. So if you want to think about a tabby cat, they're actually double striped. So they have the tabby gene that mediates the pattern of those stripes on them, but every individual hair on the tabby cat also has stripes, and that is mediated by this Agouti gene. So it affects the how tyrosine, so it's an amino acid tyrosine, how that tyrosine is modified to make the different types of melanin. So we have the black eumelanin, which is shown here, and we also have a pheomelanin that generates that ginger color that we'll get to in a few minutes. So the dark stripes of the tabby cat, if you look closely, you'll see there's no agouti patterning on those hairs. They just have an even distribution of pigment, while the background part of the tabby striping is made up of these banded hairs. So agouti controls the coding for agouti signaling protein, or ASIP, and that leads to this agouti shift phenomenon, this I guess modification of tyrosine metabolism in these pigment-producing cells, which are called melanocytes. And you see this underlying tabby pattern when there's also the tabby gene being expressed. So one really interesting thing with black cats is that if you put them in the sun, or if you have a really young black cat, you can actually still see not so much the agouti pattern, because they are they're recessive for the agouti pattern, so they are double lowercase a, but black cats still have some a ghost of the tabby striping. So they still express the tabby gene. It's just very, very hard to see those stripes without this agouti gene being turned on. So those tabby stripes need to have both the tabby gene turned on, so either mackerel or blotched, and they also need this agouti gene to be either one copy or both copies of the dominant uppercase A allele. All right, and while we're on the topic of the patterning of the individual hairs on the cat, I thought I'd bring up the Abyssinian cat. So this is a fairly fancy breed of domestic short-haired cat that has a distinctive tabby coat, and we call it ticked because instead of having just those few stripes at the end of the hair, the stripes are more narrow and more evenly distributed along the length of the hair. So hopefully you can see that in that picture there. And there are quite a few variations of this Abyssinian type as well. And it's thought that the Abyssinian originated from Ethiopia, and Abyssinia is the older name for Ethiopia. And this coat color is mediated by the ticked gene, which is shown either as uppercase TI or lowercase TI. And despite many, many hours of searching, I cannot actually find the protein expression pattern that's related to the tick gene. So I think this is really something that we're still, that's still under investigation in terms of cat genetics. So hopefully we learn a little bit more about how the ticked gene works in the coming years. Okay, so that basically covers the background color of cats. So whether a cat is black or blue or cinnamon. We've also covered the different types of tabby genes that lead to those beautiful striped cats that we're all familiar with. But how do we get a ginger cat? So the genetics of ginger cats are a little bit different than the other genes we've talked to up until now. So I've mentioned that the agouti gene, the brown gene, and the tabby gene, the ones that we've talked about so far, they're all located on autosomal chromosomes, those somatic chromosomes that are equally distributed amongst males and females. Ginger cats, on the other hand, their color is mediated by the orange gene, and the orange gene is found on the X chromosome. So this gene expresses the red pigment pheomelanin, and this has the capability of completely replacing eumelanin. So if the uppercase O gene is expressed, you will see orange fur. And if the lowercase o is expressed, you will see black or brown fur. Now, because males only have one X chromosome under normal circumstances, males can either be orange or non-orange. So you can have a fully orange cat, like the beautiful Thomas O'Malley that we have here on the screen, or you can have a cat that doesn't have any orange at all. It might be a black cat or a brown tabby cat. Females, on the other hand, have two X chromosomes, so they can express both alleles of this gene, and the alleles are co-dominant. So in a female cat, if it's a fully orange female cat, she is homozygous for the dominant uppercase O. If it is a black or brown cat, she is homozygous for the lowercase recessive O. But if she has one X chromosome with a dominant O and one X chromosome with a recessive O, she will be a tortoiseshell cat with patches of orange and black fur. And I think this is a really interesting bit of genetics that is actually kind of relevant to most of the women who are here in the talk today. So because all females are homozygous for the X chromosome, we have two copies of them. There's a lot of DNA on an X chromosome, and you really only need one X chromosome to survive. And I think all the males of our species are excellent examples of that. So because we don't need this extra X chromosome, randomly, very early on in development, a process called lionization occurs. And lionization is named after Mary Lyons, who was a British biologist. And this occurs very early on in development, and it occurs randomly. And it happens when the cysteine residues in the X chromosome get methylated, and it causes the chromosome to basically involute, to fold itself up, and to tuck itself away into the corner of the nucleus, where it is then known as a bar body. So if you have a high enough power nucleus and you have female cells to look at, you might be able to see the bar body in there. So because there is random inactivation of X chromosomes as the embryo is developing, after one cell turns off one of its X chromosomes, all of the progeny cells that develop from that single cell will express the same X chromosome. So once a bar body is formed, it doesn't come back. So that decision is made really early in development, and it's non-reversible. So essentially, this is what happens in a tortoiseshell cat, in which we have two X chromosomes here. We have one that has the uppercase O gene, and one that has the lowercase recessive O gene. So that is the allele for black. So that this chromosome will not generate that orange color. During development, the cells divide, and some cells will keep the orange X chromosome going, and you'll have orange patches of fur. And some X chromosomes will be around that do not have the orange gene, and by default you will have black fur instead. And that's how you have that beautiful tortoiseshell color. And essentially, ladies, if we could look at our skin and see the cells that we have our X chromosome from mom and our X chromosome from dad, if we were able to visualize that on our skin, we would all look like tortoiseshell cats, which I think is really, really cool. And I really wish we could do that someday. So back to the ginger tabby. So we have this sex-linked orange locus, and that leads to the production of pheomelanin, and it's mediated by the X chromosome. So there's some more pictures of some ginger tabbies. So you might be wondering about tortoiseshells and calicos. So tortoiseshells are known as tortoiseshell and white. So they are both O females, so uppercase O, lowercase O, females that express in some cells pheomelanin and in some cells eumelanin. So they're almost always female. There are some very rare occasions of tortoiseshell cats that are male, but most of them have some kind of chromosomal abnormality. So the most well understood one is called Klinefelter's karyotype, or Klinefelter syndrome. And it also exists in humans, in which that particular male has two X chromosomes and a Y chromosome. So they have the genetics to make the male, but they have an extra X chromosome, and that lionization can occur as well. And you may be wondering where these white patches come from, and we are going to get into that in just a second. Before we move on from the tortoiseshell and the calico cats, I just wanted to mention Mosaic and chimera cats. So you might hear about these cats sort of on social media or in newspaper articles. So a Mosaic cat is essentially a tortoiseshell cat or a calico cat. So there's one set of DNA from two parent cells, and because of X chromosome inactivation, the genes from one X chromosome are expressed in patches of cells over the surface of the cat's skin and fur. A chimera, on the other hand, is the result of two fertilized eggs or embryos joining to create one organism. So this is essentially two fraternal twins joining to create one cat. So the cat actually has two sets of genetics. And my favorite cat that exemplifies this is Venus the two-faced cat. If you're on Instagram, I highly recommend you follow her on Instagram. I think she's absolutely beautiful. And you can see she's got that black cat, so that eumelanin producing genotype on the left side of her face. I guess that's the right side of her face, and the ginger cat phenotype on the other side of her face, with a very sharp line right down the middle. It's not always quite so sharp on the various cats. I think Venus is a pretty special moggy for that. All right, so at this point, this is kind of where we have ended up. So we know why we have mackerel tabbies, and we know why we have classic or blotched tabbies. There are some other variations of tabbies, so you can get spotted tabbies, which are hybrids, and they're fairly rare, and they're fairly new as well. And then you can get ticked tabbies, like this one, which is the Abyssinian, and then you can get a non-striped tabby, Agouti. There are some other newer types of tabbies that have been bred and developed over the years. I'm not going to get too much into the genetics of those, and just want you to know that there are lots of variations of the tabby phenotype in cats. And this is one of those complicated Punnett squares that I mentioned before. So this is looking at a number of different genes. So ticked, so that's the Abyssinian one. Mackerel, so that's the thin tabby stripes. And classic or blotched, those are the big world tabby stripes. And then we also have black cats, blue cats, so that is the B for brown color gene, and the D dilution gene. We have all of those variations that we talked about right at the beginning. So black, blue, chocolate, lilac, cinnamon, and fawn. And then finally, we've got the ginger cats at the bottom here. So a red cat, that's kind of the geneticist term for a ginger cat, is expressing that dominant O allele on the X chromosome, whereas a cream cat has the dilution gene in double recessive, so a homozygous recessive for dilution, but also expresses the dominant O gene, that orange pheomelanin being produced by the X chromosome in those cats. So just goes to show there's a lot of genetic variation in the cats, and if you want to take a deeper dive into cat genetics, please do follow up with some of the resources I've provided you with. It's really interesting. You can go really far and find out some really cool things about your cats. Okay, so the difference between tortoiseshell and calico is the existence of white fur. So a tortoiseshell cat, you can see here on the left, has no white spotting. And we can go all the way through to almost all white cats that have a few orange or black patches around different areas of the body. So this is mediated by a phenomenon called epistasis. So epistasis occurs when one gene, the expression of one gene, is affected by the expression of another gene that is independently inherited. So they could be on completely different chromosomes. In the cat, the genes for white fur are considered to be epistatic over other coat colors. So if you have melanocytes that express either the white gene or the spotting gene, these are the two genes that lead to white color patterns in cats, that fur will come out white. And if the recessive version is there, the fur will be colored. So hopefully that helps explain the different patterns of white on these cats. But how does this actually happen? So both the spotting gene and the white gene for the longest time were considered to be completely separate from each other. But thanks to the wonders of molecular biology, we now know that they're both variations of the KIT gene. So the KIT gene in cats and in humans encodes for a receptor tyrosine kinase. So it's a receptor that sits on the outer membrane of our cells that responds to different mediators called growth factors. And both during development and during adult life, c-KIT responds to these growth factors, and this can drive cell migration. This is going to be important a little bit later on. So remember that c-KIT is very strongly related to cell migration. This white spotting can occur in many different forms. So from small white spots, so my cat Toby, who I hope you see again soon, he actually has one white toe on each of his rear paws, but the rest of him is entirely blotched tabby. And then you can get up to a cat that is almost entirely white. So the Turkish van, for example, has a pigmented tail and sometimes a bit of colored fur on its head, but the rest of the cat is completely white. If you have a complete expression of the white gene, for example, the epistatic expression of the W dominant gene, that uppercase W, you get a fully white cat. And I've got some King's Heath Cat Club examples there of these fully white cats. Now, interestingly, the white gene is also linked to both blue eyes and deafness, because you can, the cells, those melanocytes we talked about earlier, that are really important for conferring pigment to the fur, they also confer pigment to the eyes. So you can get blue eyes in a white cat, but those melanocytes can also develop into some sensory neurons. And because of this deficit in how these melanocytes move during development, you can actually get deafness in these cats. So a large number of white cats with blue eyes are deaf because of their expression of that dominant uppercase W white gene. So here's a really bit of interesting stuff on the spotting gene. So this is the gene that leads to incomplete whiteness on a cat. So you can see this black cat here is a non-white spotted. And then as we move further to the right, we get a higher grade of spotting. And the pattern of the spotting can change as well. So we've got one cat here with mittens. We've got a tuxedo cat here. We've got a mask and mantle cat, this bicolor cat. And then you get to the Turkish van is over here with that black tail and a bit of black on its head. So you can see there's a high degree of variation in how that spotting gene is expressed. And I'd like to just show you as an example, my cat Darwin, who is decided not to participate in today's talk. He's sitting on the windowsill behind my computer, thinking about whether he's going to join us, I think. So you can see Darwin, similar to Toby, is a blotched tabby, but he has also widespread expression of the spotting gene. And you can see that his tummy is mostly white. He's got a little bit of a tuxedo jacket that meets in the middle. And he has white socks on all four of his paws. And he also has, you can't really see it in this picture, but he's got a white flash on his face. And that is actually why he's named Darwin, because that white flash on his face kind of looks like a beard. And he kind of looks like Charles Darwin on the old tenor. And that's why we called him Darwin, also because I'm a nerd and I love biology, and Charles Darwin, I think, is a really cool biologist. But yeah, there we go. That's why Darwin is called Darwin, because that white flash on his face from his spotting gene makes him look like Charles Darwin on the old 10-pound note. So there are a lot of theories as to why these patterns arise. And I'd like to highlight that this is very, very much under investigation. So there's quite a bit of controversy in the field as to which one of these possibilities is really legitimate. It's somewhat difficult to study development in animals that we love, because it usually involves the sacrifice of a viable embryo. So in any case, similar to what has been observed in the mouse and the chicken, the cells that produce pigment, as I mentioned before, are called melanocytes. They produce melanin. And they migrate during development from the neural crest. And they move from the center of the body at the back, and they move down through the body, so along the sides. And then as the limbs develop, they move out to the distal ends of the limb. So you can see these melanocytes in red here, and they're following a gradient of different signaling factors and growth factors as that embryo is developing. So one theory is that in a cat that looks like this, a cat that looks like Darwin, these melanocytes just didn't make it to the last parts of the body to develop very early on. So you see the embryo is starting to develop, these melanocytes are starting to migrate, and they just never make it to that center line of the body. Another theory is that the melanocytes do make it all the way, but something that we don't understand yet is causing the melanocytes to die around the center line of the body. So two possible explanations for why we get patterns like Darwin's. And then in this case, which is still not very well understood, we get random patches of pigment and white patches on the cat. And yeah, we're pretty sure it's because of melanocyte migration. I had a really good chat with my colleague, Dr. Anne Vernalis, about this yesterday. But again, there's not really a huge amount of solid scientific evidence for this in cats. So again, watch the space. I think there will be some interesting work that happens in this particular area. So just to showcase some of our beautiful tuxedo or black and white cats that we have in the King's Heath Cat Club, and some of our tabby and white cats there. So you can see Darwin's face a little bit better in that picture. And just for everyone to appreciate the huge amount of variety and beauty that we get from the spotting gene in these cats, whether they are recessive for the agouti gene, so that'll be these tuxedo and black and white cats that have that black fur, or whether they have at least one copy of the agouti gene and look like tabby cats. Okay, so now we can move on to some of the more exotic cats. So cats that have arisen, in the last, I'd say, 100 years or so. So a really well-known one is the Siamese cat. The Siamese cat is mediated by the color gene, and that's given as the symbol C. So there are a couple different variations of this, but these cats end up being called color-pointed, and they have darker colors on their face, ears, feet, and tail, and a lighter version of the same color on the rest of the body. And like most of these cats that we've talked about today, there's a huge variety of the different types of color points of Siamese and Burmese cats. So the dark brown one is called seal, for example. And the reason why we get this pattern in Siamese and Burmese cats is because of a mutation that causes tyrosinase to be temperature sensitive. So this is an enzyme that helps convert tyrosine into the different types of melanin, so whether it's eumelanin or pheomelanin. And if tyrosinase has this mutation, it's temperature sensitive and it stops working in the warmer parts of the body. So you get less pigment in the warmer parts of the body and darker pigments on the cooler parts of the body, and that's the face, the paws, and the tail. So the uppercase C gene is what we see in most cats, so that's full color. The C with the uppercase B is a Burmese or sepia pattern that's somewhat lower contrast. You can see that in fancy here. And the CS or Siamese variant has the stronger color variation that Lola is doing a great job of showing us. Interestingly, the fully recessive lowercase c generates an albino cat, which is very, very rare. But there is also an association with this gene and eye color. And we'll get into eye color a little bit later, but often, as Lola is showing, Siamese cats have blue eyes. Bengal cats are another type of exotic cat, and they're actually a hybrid of domestic cats, mostly the Egyptian Mau and the Asian leopard cats, so Prinoloris bengalensis. So they're not fully Felis catus. They're a hybrid of two different species. So they have a fairly wild appearance. Hopefully Enzo and Roxy are showing you that pretty well. Their golden shimmer comes from their leopard cat ancestry, and their fur comes in really, really interesting and beautiful patterns. They're a very energetic breed, and they need a lot of exercise. And unfortunately, because they are a hybrid, there can be problems with sterility. So a lot of times hybrids, so if you think of a mule, which is a hybrid of a horse and a donkey, oftentimes they are sterile. So obviously this doesn't happen all the time in Bengal cats because people are breeding them, but it does happen from time to time. So this breed arose in California in the 20th century from Gene Mill. But now let's get into hair length. So there are lots of interesting variations of cats that have long hair. So there's a gene called length in which the dominant form, uppercase L, encodes for short hair, and the recessive lowercase l encodes for long hair. And this gene has been identified as fibroblast growth factor 5. So we've talked a lot about different growth factors today. So this is the particular growth factor that the expression of this growth factor changes with that recessive lowercase l. And it means that the transition from the growth phase, that anagen phase of hair growth, to when the hair stops growing, that catagen phase, it's slowed down. So the hair grows much longer than we would see in a cat that has the dominant version, that uppercase L, that would have short hair. So there's at least 4 different mutations in the L gene that have led to this phenotype. So Persians, of course, have a mutated recessive L gene, but this gene is also found in ragdolls, Norwegian forest cats, and Maine Coons. So Persian cats are probably the first fancy cat, if you want to think of them that way, the first show cats. So they're a long-haired breed with a round face and a short muzzle. So Ipsy is showing you that there, and there's a historic picture of a Persian cat from 1894. So back in the 19th century, that's when the first Persian cats really arose. So they were actually first started in 1620, but it wasn't until the late 19th century that they really came into being as a specific breed of cat. And eventually, after World War II in particular, in the UK and in North America, they became incredibly popular. So there's been a huge amount of breeding of these Persian cats. So cat fanciers are aiming for a variety of coat colors, but unfortunately, they're also trying to get the face to be a little bit flatter, and that can lead to a lot of problems with how the cat breathes and eats. So hopefully there is a bit of, I guess, movement in the cat breeding community now to try to think about the well-being of the cat as well, and maybe not go for these extreme phenotypes that we see with these short-muzzled cats. Also important to remember that Persians have a genetic susceptibility to hereditary polycystic kidney disease. So if you have a Persian cat, please keep an eye on how much they drink and get their kidney function tested by the vet. Hopefully your vet is on top of that. Other long-haired cats include Maine Coon and Norwegian forest cats. So it's thought that the Maine Coon developed from the Norwegian forest cat. So they're very close in genealogy, very closely related to each other. The Maine Coon obviously was first bred in the American state of Maine. So these are very large cats. They have a very distinctive physical appearance. They are incredible hunters, and they're very, very sociable and very intelligent. And we are pretty sure, I really wish I could show you right now, we're pretty sure Darwin is about a quarter Maine Coon, because if you can see on the lovely Rufus here, also known as Lord Rufus, he has black tufts on the end of his ears, and Darwin has those as well. And I think maybe he's definitely not full-bred Maine Coon because he's a short-haired cat, but he has those tufts on his ears, suggesting that there is some Maine Coon in his ancestry. So for a long time, they were popular, like in the 19th century, and then the Persians really kind of came in and got very popular, but the Maine Coon in particular has had a resurgence in recent years and is now the third most popular pedigreed cat breed in the world. And I just thought I'd quickly cover ragdoll cats. So there's not really a specific gene that mediates ragdoll cats, from at least from my research. It'd be interested to find out if this does come up in the near future. But the ragdoll cats originated in California in the 1960s, and they originated from one particular cat named Josephine, who happened to produce litters of cats that had very long, soft, and silky coats. But when you picked up the kittens, they just sort of went limp. So that's why they're called ragdolls. So these cats are really, really docile, very placid, they're very affectionate, and as you can see, they're also quite beautiful. So Kings Heath Cat Club has some ragdoll cats with Cinder, who's the mum, and Charcoal Bear, who's the baby. And these two cats, Bingley and Darcy, I think they're just absolutely gorgeous. I follow them on Instagram as well. So if you're into ragdoll cats, you might want to look around on Instagram for different ragdoll cat accounts. And going the opposite way, we've got the Sphinx cat. So this cat, either spelt with an I or a Y, I've seen both. It's known for its lack of fur. And as you can see, this particular cat, I found a picture of this one on Wikipedia. He's got a little hoodie on because he doesn't have any hair. And if that cat's living in a cold environment, it's going to be chilly. So ironically, these cats actually were developed in Canada. Let me tell you, winter in Toronto is not warm. So hopefully these cats were well looked after by their humans to keep them warm in the winter. So they started in the 1960s, and there are breed standards for this cat, so they should have leather-like skin with fine hairs or be completely hairless. They may or may not have whiskers, and I invite you to look up a picture of the paws of Sphinx cats. They have long fingers with webbed toes. Very, very interesting kind of cat. Interestingly, with these cats, the founder population of these cats, the ones that start in Toronto, is very small. So there's been quite a bit of inbreeding in the Sphinx cats. So that's something to keep in mind if you do have Sphinx cats, is that they had a very small founder population upon which the breed was developed. And now we're getting into some of the variations of cat body type that can be a little bit detrimental. So the Manx cat, so I've shown here a historical photo of a Manx cat from, when was this? From the early 1900s in the UK, named Silverwing. These cats originated on the Isle of Man, so they're called Manx cats. And the gene that mediates the Manx body type is called Felis, or Manx, and it's symbolized by the letter M. So uppercase M is dominant and will lead to this particular genetic mutation or variant, and the lowercase m is recessive, and that's the variant of the gene that most cats have. So cats that have tails have a double copy of the lowercase m Manx gene. So this gene encodes for T-box transcription factor, which leads to the premature truncation or cutting down of the brachyury protein. So this brachyury protein is really, really important during development for establishing the anterior-posterior axis of the embryo, so the head to tail axis. So hopefully you can see now why a problem with brachyury leads to the loss of a tail. So also interestingly with this cat, the dominant form in its homozygous form, so a cat that has uppercase M on both alleles is lethal. So those cats, if they are generated in a breeding program, the embryos just never develop and the kittens are never born. So all the Manx cats you see are actually heterozygotes. So many Manx cats will have a small tub of a tail, but many of them are entirely tailless. And their coat patterns are, generated by the other genes that we talked about earlier on in this talk. So we're just specifically talking about the body structure in these cats. And unfortunately, this dominant uppercase M allele can have some really serious health implications for the cat. So it can lead to lameness, incontinence, and constipation. And again, because that homozygous uppercase dominant M is lethal, breeding these cats can be quite tricky and really should only be undertaken by someone who really knows what they're doing and knows how to take care of these cats with all of their inherent health problems due to this genetic variation. And finally, we've got the Scottish fold cat. So why these cats both developed in Britain, I don't know, but the Scottish fold cat first arose in 1961 in Perthshire. And it can either be a long-haired or a short-haired cat. It can have any variation of color based on all of the genes we talked about earlier, but in particular, these folded ears. So you can see these folded down ears that kind of tip forward, kind of make the cat look like an owl. That is mediated by the fold gene, and that's shown as the dominant version uppercase FD and the recessive version lowercase FD. So similar to the Manx cat, the gene that causes the ears to fold is inherited as a dominant trait. So one copy of the fold gene will give you these folded down ears. It's also suspected that homozygous dominant for the uppercase F fold gene are lethal as well, similar to the Manx cat. So again, one of those issues with breeding that need to be considered if you want to get into these kind of cats. Now, the protein that is mediated by the fold gene, so what happens there still isn't really well understood, but it's thought to lead to a defect in collagen production, and that can then have carry-on effects on how cartilage is formed, and that's the cartilage in the ears that is malformed in some way that allows those ears to tip forward. Similar to the Persians, there's a high level of polycystic kidney disease in Scottish fold cats. There's a higher level than usual of cardiomyopathy, so problems with the heart, and there's problems with the bones and the cartilage as well. And I think that is specifically related to that dominant fold gene that probably has some effect on one of the many types of collagen that are in the mammalian body. So that's about it in terms of what cats look like in terms of their fur and their body shape. And I thought I'd just put one slide on here about eye color. So I talked about earlier with the Siamese cats that there is a relation with the blue color of eyes related to that Siamese variation of the albino gene. But most cats have eyes in this kind of color range. So you go from green all the way to copper. So Darwin has green eyes, Tiger had golden yellow eyes, and Toby, my cat, has copper eyes. And to be honest, other than that one gene variant related to Siamese coat color, we don't really understand the genes that mediate eye color in cats. And to be honest, we don't really understand how it works in humans either. It's definitely polygenic, so there are many different genes that are involved. One layer of complexity is that there are two layers of pigment-producing cells in the epithelium of the iris. So there could be, multiple genes and multiple cell types involved in leading to these different ranges of eye colors. So hopefully we'll be learning a little bit more about the eye color genetics of cats in the coming years. One thing I can say is these odd eyes. For example, a golden eye and a blue eye on opposite sides of the face. This is most often found in cats that have that epistatic uppercase W white gene, in which one eye is blue and the other is a different color. So it tends to be white cats that have odd colored eyes. And I thought I'd also throw this one in here, polydactyly, or cats with thumbs. So this is mediated by the same gene that causes polydactyly in humans, called polydactyly. And this does have some variable gene penetration. So you can get cats with 1 extra thumb or 2 extra thumbs, and it can get really, really interesting. And like in humans, polydactyly encodes for the sonic hedgehog gene. So I really love the name of this gene, sonic hedgehog. It turns out if you knock it out of a fly, it makes the surface of the fly look really, really spiky, and that's why it's called sonic hedgehog. But in mammals, sonic hedgehog has some really key roles in cell growth, cell specialization, and importantly, when we're talking about polydactyly, the normal shaping or patterning of the body. So this is the same genetic variant that can occur with humans with extra fingers. So hopefully everyone who's watched The Princess Bride remembers the 6-fingered man. And these extra digits also vary in number and structure. So I've got a picture there of a cat with 2 extra toes, 2 extra thumbs there. And one of my favorite cat memes, this poor cat with thumbs is trying to text his friends because he has thumbs, but his other cat friends don't have thumbs and they cannot text him back. Very, very sad. And I'd just like to finish off today's lecture with a bunch of memes about cats. So I think cats are generally well loved. I mean, especially in Britain, British people, you just love your cats so much. And they're such a force in, I guess, social media and just generally what we like to laugh about. So Icon has cheeseburger. That one's been around for a very, very long time. I think everyone's familiar with all the variations on this one as well. And I especially love the creation of man with a cat in this place. And yeah, so hopefully everyone's enjoyed the lecture on cat genetics today. I'll just quickly put up my references. So I will try to get these made available to you, possibly after this talk gets put up on YouTube. I'll put these into the comment section of the YouTube link. And hopefully if you want to follow up on some of the information I provided you with today, please go ahead and do that. So there are some, for the more lay audience, a couple newspaper articles that you might want to look into. And if you want to really get into the science of this genetics, here's the list of papers that I used as my sources for this talk. And finally, I'd like to acknowledge, of course, my beloved fur babies, Darwin, Toby, and Tiger. If you'd like to follow my fur cats on Instagram, they're at Darwin McBraveheart. And of course, I'd like to thank my husband, Darren, who has been the best cat dad and is also very, very supportive of me just in general, but has been really supportive with this talk. And Dr. Zita Balklava, of course, for moderating today's talk, and Dr. Anne Vernalis for her intellectual contributions on mammalian development over lunch this week. And again, King's Heath Cat Club, thank you so much for all the pictures. And again, I'd like to thank the West Midlands branch of the Royal Society of Biology and the Shrewsbury Darwin Festival for inviting me to give this talk. And again, those are all of the pictures of cats I got from the King's Heath Cat Club from one single request. So they definitely stepped up. So thank you again for all of those pictures.